All right, we're back with part 2 of this little mini sequence. This time we're going to talk about measurement. Again, just to provide an overview of some of the measurement techniques that we use in the lab, and a little bit maybe clinically, but mostly in the lab, to measure tissue behavior. Keep in mind that we have this underlying picture of cells coupled to cells, and that our measurements have to happen in that extracellular space. So you can picture these as mostly voltage measurements, either directly or indirectly. And they require 2 electrodes, like all voltage measurements do. So we can place those electrodes, and they come in lots of different forms, in that extracellular space. So key point here, all measurements at the tissue and whole heart scale are made in the extracellular space. We don't stick electrodes into the cells, the individual cells, to measure transmembrane potentials like we do at the cellular scale, except in some very unusual circumstances, very artificial and very experimental situations. So most of the time, overwhelming majority of the time, we're measuring extracellular potentials, and we'll see what that means in a sec. Now, one of the measurements, I should say, is kind of a structural measurement in that it's possible using a technique called confocal microscopy that you may have come across that allows us to measure the cardiac tissue at different depths. Now, here you see a schematic of it's more complicated than we need to get into, but this was a technique developed actually at our university at the CVRTI by my former colleague Frank Saxe. And Frank and his students developed this method to get very clean, very structurally accurate measurements, optical measurements in this case, using confocal microscopy of this tissue structure. And the interesting thing here is they're able to take superficial measurements on the epicardial surface and then move down through the tissue in steps and ultimately reconstruct 3-dimensional tissue structures. And here you see a picture they made of individual myocytes in different colors here, bundled together to make a small slice of myocardial tissue. Again, that slice has a clear orientation, but if we moved all the way or deeper into the tissue, we'd find that slice orientation would rotate. That's that anisotropic conductance, anisotropic structure that also is rotating anisotropic structure, just to make things complicated. So that's sort of a structural measurement. We want to get back to functional measurements in this case, and electrical measurements are certainly the most common in many settings, although we'll see shortly, there's optical approaches, not surprisingly, they're also very important in research. So the idea here is that we can place electrodes on the surface of the heart. We can place, and we'll get into this a little bit more in the next week, we can place electrodes on catheters inside the heart in animals and in humans. This is done clinically all the time, and we'll come back to that. Only in the lab, as an experimental method, we can also create electrodes that impale the tissue, that have multiple individual electrodes along what we call a needle, and then that needle gets inserted into the tissue and will stay put, and this will allow us to make 3-dimensional measurements. So surface measurements are 2-dimensional, of course. With these needles, multiple needles like this, we can actually create 3-dimensional measurements. And these are the tools of the trade that we use to capture cardiac electrical activity. We'll focus mostly on the surface ones for this course. And here already you see a rendering of one time step, because this is a wave that's moving. And here we see a voltage map, this is called. And the dark blue region is the region of negative potentials. That means that it's the location at this time point where tissue has been activated. It may seem counterintuitive that you have negative potentials where things have been activated, but this does make sense if you dive deeply enough into it. The point is that we have a spread of activation, and we have those elliptical, sort of bent elliptical lines. The same thing we predicted based on the structure we actually see in measurements. So the blue region here is low voltage, yellow-green mid-voltage, red is higher voltage, actually 0 in this case. The whole voltage range, as you see in that scale, plays out between 0 and minus 30 millivolts. So we have a pattern in space that evolves with time. And I'll show you an example of that just shortly. Once we have voltage, we can also calculate, literally, from the voltage gradient what the current pattern is. The current and voltage are related through the gradient operation in 2-dimensional space. This is another form of Ohm's law. And so we're able to draw lines because current is a vector. It flows in space, so it has a vector quantity, has a direction. And we can draw these lines, we can draw flow lines, we can do all sorts of interesting things with voltage data, voltage signals that we've captured. But we need multiple electrodes. You can picture this two-dimensional picture came from multiple electrodes all capturing simultaneously. And as I say, that also evolves and changes with time. So you see the role also of anisotropy here. Now, of course, there are optical equivalents, just like I mentioned in the cardiac cellular space. We can shine laser light on a heart to excite. We can apply a fluorescent dye that fluoresces as a function of the membrane voltage. So this is one way in which we're actually measuring local membrane voltage, even though we're not sticking electrodes into the heart. And this is the clear advantage of these optical methods over electrical measurements, is that we're getting something that is more directly linked to the underlying cellular behavior. Resolution is a question and a limitation, resolution in time, resolution in space, all these various aspects that arise in any optical method. But this is an often used approach that we see in the literature, especially again in experiments. So we're able to stimulate with a light source, we're able to record the fluorescent information on a grid and piece together a two-dimensional map that evolves with time of that underlying activity. And here you see an example of the activation time. So this is not the voltage itself. This is like the peaks of that water wave, if you picture. You throw the rock in the water and you look at the high points of the water, the ripples. And if you put all those ripples together, you can get a map that looks like this top map here, which has activation time. So blue is early time, red is late time. And again, you see that elliptical pattern of the spread of activation driven by the structural underlying relationship. And this plays out on a very small scale. Here you see this is a rat heart, which is about the size of kind of the tip of your baby finger. So these are very, very small hearts. And it's possible to measure those using optical techniques and to measure individual signals that are also in this figure that are really direct renderings of the underlying membrane potential. So you can see the equivalent of an action potential measured at these sites, and then from that action potential timing we can create the activation maps. Below there's an action potential amplitude map as well. We often want to know spatially, is there a distribution of amplitudes of the underlying action potentials? Depending on the question at hand, that can be very important. Now electrically, we have two different basic forms of measurement. We'll tell you a little bit about these. They do arise in both clinical medicine and in the lab in experimental settings. One is called a unipolar measurement. And what we have in a unipolar measurement is a single electrode that's sitting somewhere near the tissue, somewhere near the region of interest. And its measurement is taken relative to some remote electrode. Now, remote can mean lots of different things. We'll see in ECG the same concept arises, but remote is thought to be some electrically neutral area away from the heart, or sometimes literally away from the heart. And so we see a signal that is very sensitive to local activity, but also picks up more remote activity. It's sensitive to whatever's going on around it broadly. And it of course generates a certain shape from which we can extract relevant parameters. And here you see a sequence, 3 images in time that show the wave front, the excitation front moving toward the recording electrode. As it moves toward it, you see the voltage will go up. As it passes, that voltage will go down sharply. So again, that negative voltage when we're in the region of activation. And then it returns from that negative value as the wavefront moves away from it. And those are characteristic features that we see in a unipolar electrogram. Bipolar electrograms, as the name would indicate, involve 2 electrodes closely spaced together. These, again, are used clinically quite a bit, also used in the lab. Here we have 2 electrodes close together. The difference between the voltages sensed at those 2 electrodes is what is the signal that we collect. And in this case, we have a rapidly changing signal as the wavefront passes by, But then when the wavefront is either ahead or behind that sample region with the two closely spaced electrodes, the difference between the voltages in that case is very small. So these electrodes are very insensitive to remote electrical activity and only really turn on when the wavefront is passing by. And that has certain practical advantages and is the motivation for doing it. The downside is that the wavefront is much harder to interpret and can change its shape quite dramatically depending on how the wave is moving relative to the orientation of those two electrodes. You can kind of picture this. And so again, they're useful and they're used to determine this activation time, this time when the wave passes that location, but they're less useful when it comes to interpreting the shape of the actual bipolar signal. Now here's an example. So here we have an ECG in the top row, and then we have unipolar electrodes, two of them, and then we have the bipolar electrode that results from subtracting those two electrodes. So the unipolar is called M1, M2, and the bipolar is M1 minus M2. So we've constructed a bipolar signal from the two unipolar signals, which is totally fair game. The two unipolar electrodes are pretty close together. They do have very similar signals, as you can see there. You can see there's activation that aligns with the bipolar activation, but then there's another big deflection that occurs later in the signal, and that, if we look at the ECG, corresponds to the big spike in the ECG. Now the bipolar electrodes become insensitive to that activity because these electrodes are actually placed in the atrium, one of the atria. And so the signal is locally sensitive to the atrial signal, which is quite small in amplitude. It corresponds with what we know, we'll soon learn, as the P wave. That's the wave that initiates the ECG signal for a single heartbeat. And we see that small P wave on the ECG translates into a pretty good sized signal in both the unipolar electrograms and then especially in the bipolar electrogram. So the bipolar is very sensitive because it's sitting in the left atrium in this case. It's very sensitive to that left atrial activity, but not sensitive to that remote ventricular activity because both electrodes sense it with the same shape and the same strength. So there you see, in one practical example, how we can use these two electrode systems. Now in engineering, when we have a signal, we want to process it. That's kind of a reflex we have, right? We got to clean it up, got to get rid of noise, we got to filter it. There's all kinds of useful things we can do with it. And of course, in this particular context, there are some cardiac specific indices which we can also measure. And we'll, again, discuss some of these, I think, in the discussion session. But what we have here is a typical, what we call an electrogram, which is different from electrocardiogram in that electrograms are typically recorded directly from the heart or near the heart, and electrocardiograms are recorded on the body surface. So here we have an electrogram. It doesn't look like an electrocardiogram, as we'll see, but it has these same letters we sort of use. That we will learn about next week. It has these QRS letters here. But what the key feature is, as we saw before, is when the wavefront is moving toward this unipolar measurement, then we see an upstroke or a positive deflection, that's the R wave, and then we see a rapid downgoing deflection until we get to the most negative part, that's called the S wave. The time of that maximum downstroke is the activation time. Then the signal returns, spends some time sort of fairly flat in the middle, and then there's a positive peak, and that positive peak corresponds with the repolarization of the action potentials in the nearby tissues. So this tissue is an integral of lots of action potentials. It's extracellular, so it's an indirect reflection, but it still captures those key behaviors of depolarization, the upstroke of the action potential, and repolarization, the downstroke of the action potential. And we can capture that information, or pretty accurate rendering of that information, by looking at the activation time, the maximum downward slope, and the recovery time, which is the maximum upgoing stroke or upstroke of the electrogram in that region sort of near the end. And so these are features we can pull out of electrograms and do quite routinely and have software to do all that. And we'll talk about some of the algorithms that one might use for that. We also see this baseline. In reality, those baselines tend to wander. We don't always have a perfect flat baseline, so we have to correct for that. So it's another trick, if you will, in the arsenal of the signal processing of these kinds of signals. And as I said, the unipolar electrograms contain information in the shape. So if we observe the shape, we can tell things like when the wavefront passes. We saw that, the maximum downstroke. We can say something about where the electrode's located relative to the wavefront throughout the heartbeat itself. And here you see an example, just one simple example. On the left, you see a map of the voltage recorded over an entire left and right ventricle. So here we have a lot of electrodes, 500 electrodes, it turns out, in this case, wrapping around the ventricle of this animal heart. And the blue is, again, the region of lower potential, more negative potential. That's the region that has been activated. The yellow is the sort of middle region, it's around zero, or where the wavefront is passing. And then the red we see further away, the more positive, that's where the wavefront has not reached yet. So this elliptically shaped wave is going to be moving away from this site in the middle there where the top arrow points. And now we see the individual electrograms, and we see they have different shapes, very clearly different shapes. And that tells us something about where they are relative to the initial site of the wavefront, where that beat was initiated, in this case at that sort of middle point where the yellow dot is. And we see that that's the case because the positive going part of the signal is very short. So that electrode is located close to the site of activation because there's only a little bit of time when the wavefront was moving towards it, and when it moves towards it, you get a positive deflection. Then it passes, and then it moves on. The second trace down is further away. This is sitting pretty much at the point where the wavefront is about to hit it, and you can see it's been positive for quite a while. So it stays positive for a lot longer before the wavefront passes it. Large downstroke, that's the activation time, then it moves on. And finally, the third tracing is a point quite away from the actual activation site, the location where the activation starts. And there we see the longest duration of positive signal because it has to wait for a long time for that wave to actually reach it. It does have this sharp negative downstroke. That marks where activation happens or when the wavefront passes by that location. But its negative region when it's passing away is also quite short. So this is an indication that this electrogram was recorded far away from the site of activation. And so we can see this wave shape can tell us something useful, and it is used clinically to indicate where waves are coming from in a complex pattern in a heart. Here we see the last image I want to share with you, which shows a sequence of 4 individual maps. So the 4 figures on the left here are all individual time points. So here sort of capturing in time step the initial site of activation, that little blue core at the middle there, and then it spreads out going to the right, becomes elliptical with sort of a narrowing, and then the lower left image shows it spreading in a more complex shape, and then finally we see what the pattern of voltage looks like near the end of the wave. You can see the white line in the time tracing below tells us where we are in time through the heartbeat. So we're seeing 4 snapshots out of an entire heartbeat, and you see there's quite dramatically different shapes that arise through that beat spreading. But the idea is negative regions or more negative regions are the regions activated, and we can watch that spread. On the right-hand side, we see another example of one of these activation maps. So the top right map is an activation map. So here the color corresponds to time value. And there again, you see that the low time numbers, the 0 time, that dark blue, that's sitting in the middle at the same spot as we saw in that upper left image as the site of origin of the beat itself. And this captures the entire shape of the wave as one beat goes through. And then down below, we have the corresponding complementary recovery time. So that's the time at which those action potentials are repolarizing or the tissue's recovering. So that gives you at least a little bit of an insight into how this measurement process works. This is, again, there's lots more depth. We have a whole course in which we dive much more deeply into these questions. But these really are the techniques that we use in the lab, and then derivatives of those, as we indicated, catheter-based approaches, they're used in clinical medicine, and we'll come back to those when we talk about the whole heart. So hopefully that gave you at least a quick view. Look forward to seeing you soon, and thanks for your attention.